Alright, so we're going to start up with our first lecture here on the acoelomate and pseudocoelomate invertebrates. So, in the last chapter we talked about parazoans, and we said parazoans is synonymous with the phylum Porifera, which are the sponges. And of course the thing that makes them different from all the other animals is the fact that they don't have tissue level structure. Now they do have some very, very basic specialization, but really not so much that you would call them tissues. And we'll really kind of clarify what that means here in just a few minutes. So sponges are kind of our first step here in this chapter. Now sponges, because they don't have tissue level organization, also don't have symmetry, it doesn't really make sense to classify them as protostomes or deuterostomes. We technically can call them acoelomates, although it's kind of a strange way to consider them acoelomates. They still are that because they don't have any form of a body cavity whatsoever. The adult form is sessile, and so first time we've seen that term, sessile means they're attached, and they're stationary, they don't really move around. The juvenile form, though, is motile, and motile is the opposite of sessile, so it means they're able to move around. Motile and mobile are kind of the same thing, not exactly, but pretty much the same thing. So the juvenile form is motile, so they do classify, or they qualify as having movement during some stage of their life, as we talked about in our previous chapter, even though the adult form is stationary. Very little organization, like we talked about. And two structures that are unique to sponges are spicules and spongin. All right, so first off, spicules are very, very sharp barb-like structures found within the body of the sponge that helps to give the sponge really two things. Number one, it's structural support. All right, so this is where most of the integrity of the sponge lies, is within the spicules. And also defense, because spicules, like I said, are barb-like. They're typically made out of either silica, which is essentially like glass, or calcium carbonate, which is like limestone, but in this form, very, very sharp barbs of limestone. So they're also a defensive structure as well. We don't really find spicules in any other organism except sponges. And the other thing we find in sponges is a protein called spongin. And so spongin is kind of the squishy stuff that you think of. When you think about a spongy texture, what you're thinking about is spongin, right? So that's basically what gives the sponge its overall texture, is that spongin protein within. So this is the first time we've looked at a phylum now as a group. So a couple of things I need to mention here. Number one, I'm never going to ask you guys exactly how many species are in a group. Right? So I'm not going to say, okay, how many species of sponges are there? How many freshwater sponges are there? But what you do need to understand and what you do need to know is, relatively speaking, how big is the group? So 7,000 species, we don't really have a point of comparison yet, but as we're going to see, 7,000 species in a phylum is really not very much. This is a pretty small group. It's not the smallest group that we're going to see, but it is among the smallest groups that we're going to see at the phylum level. All right, so remember our classification, taxonomy. It goes domain is the highest level, the most general level. And then we have kingdoms. Of course, we're in kingdom Animalia for most of the first part of what we're dealing with right now. We have phyla. Phyla is plural for phylum. And then we have classes, and we have orders, families, genus, and species. So we're at a really high level, dealing at phylum level. It's a very, very broad, very general, fairly inclusive group. So 7,000 species in a group is really not very many. And 150 freshwater species is also a very, very small amount. So another kind of thing to clarify here. Whenever you see SPP like this means more than one species. If you just see SP, that means single species, right? So there really isn't such a thing as a species. You'll see people use the term species from now and again. That really isn't a term. It's a term people use, but it's not officially scientifically a term. Species can be both singular and plural. You can have one species, you could have 10 species. It's kind of like using the term deer, where you could have one deer, you could have 10 deer. You don't have a D, right? So species can be one, or it can be 1,000. It's both singular and plural. So when we abbreviate that, SP means one, SPP means more than one. All right, so enough of that. Back to sponges, who are among the most abundant animals in the deep ocean environment, which is actually a really handy place to be able to live if you're an animal, because the deep ocean environment is very, very, very stable. Right? It's a fairly harsh place to live, lots of pressure, fairly cold, not a ton in the way of food, you might think, but it's also very stable. right? So if something happens on land, even something really catastrophic like an asteroid slams into the Earth, like what ended the dinosaurs, or most of them at least 65 million years ago, if you're living in the deep ocean environment, you really may not notice a change at all, right? Because it's already dark, it's already cold, there's already a lot of pressure, and those things aren't going to change just because something dramatic happened on land. So if you're an animal that can live in a deep ocean environment, it's actually a fairly stable and protected place to be able to live for animals that can eke out a living there. They are filter feeders, which is how they make a living in the deep ocean. They essentially bring in a water current. They take whatever food particles might be in the water and they digest those. We'll talk about that here shortly. And then they expel cleansed water, essentially water that's been filtered back out the top part of the sponge. This opening up here is called the osculum. The places where water goes in are microscopic, essentially. You can't really see them in this picture, but they're going to go in through the side of the sponge, and then water is going to come back out the top of the sponge through the osculum. And along the way, whatever food particles are in the water are filtered out and digested by the cells of the sponge. So if you're living in the deep ocean, you got all kinds of dead stuff raining down from above. There's always going to be food coming down to those deep ocean environments, and if you're an animal that can filter feed, that's a pretty good way to make a living. We talked about the fact that larval sponges are free living and the adults are sessile. There are lots of different growth forms of sponges. A lot of times we'll think of things like these azure tube sponges or the yellow tube sponges we saw on the last slide. But there are all kinds of other sponges, like encrusting sponges that kind of grow as like a layer on the bottom of the ocean. There are barrel sponges, which are like this, but much, much bigger. Lots of different variation in the colors and shapes and sizes of sponges. I've seen them scuba diving. Barrel sponges that are this, big enough for you to crawl inside of if you really wanted to. I've seen barrel sponges that will have nurse sharks sleeping in them, like pretty good size, 4-foot long shark curled up asleep in a vase or a barrel sponge. They can be pretty big, but they can also be very small as well. As far as cell types, we'll talk about those here shortly. There are essentially three different kinds of cells. There are those that live on the outside of the sponge, epithelial cells. There are collar cells inside the sponge, which are the majority of the cells and the ones that do most of the work for the body of the sponge itself. And then there is one other cell type that we'll talk about as well as we move along here. So this is a pretty good cutaway view of a sponge, which kind of shows those really important kind of collar cells we talked about a second ago. So we have pores, or openings, hence the name porifera, openings in the side of the sponge where water comes in. And the water comes in because of these collar cells moving their flagella. So as these collar cells all move their flagella, that creates a water current, and water is drawn in through the pores. Food particles are filtered out by these cells. And then the cleansed water is pushed out the osculum up here on top. Right? So these cells, once they're brought in the water, every single cell is responsible for capturing and digesting its own food. So really not a lot of cooperation here. This is the only example of an animal that feeds this way, where every single cell in the animal is responsible for capturing its own food. Our bodies don't work that way, right? If we bring in food through our GI tract, our brain cells don't have to capture food and eat it from our stomach, right? We break down nutrients in our GI tract, they're absorbed in our small intestine, they go into our blood, they're processed by a liver, and eventually those nutrients make it to our brain cells, which then have a supply of energy. But the brain cells themselves don't have to capture food particles. If you're a sponge, on the other hand, every single cell has to capture its own food, digest its own food, absorb its own nutrients, and make a living that way. So even though they cooperate by creating a water current to bring water in, when it comes to eating, every cell is kind of having to fend for itself when it comes to getting nutrients and breaking those down. And then we see a few other structures here in the cutaway view. We have some spicules here. We have some spongin. Spongin is kind of like that protein that just sort of holds everything else together. The collar cells, the epithelial cells, the spicules are all kind of glued together by the spongin protein. Right? So collar cells, because they have a ring of essentially like cilia around the outside, which looks kind of like a collar, which surrounds this flagellum. And these are very similar to those choanoflagellates we saw at the end of the last section, which are proposed to have given rise to the rest of the animals. Pretty good evidence here. These look just like individual collar cells that make up the body of the sponge, so it's not surprising then that we think animals probably came from a similar kind of organism. So even though we don't really have true specialization, there is a degree of colonial nature to sponges. They are considered to be multicellular enough to fit in with the animals because they have that cooperation among, for example, the collar cells bringing water in that way. That's a degree of cooperation that you wouldn't really see if it was strictly a colonial organism. The contraction of the osculum, that opening at the top of the sponge, water can be forced out more or less forcefully depending on the needs of the animal. So that's also a degree of cooperation if those cells contract. There is a degree of cooperation and synchronization when it comes to reproduction, because as you might imagine, if you're an animal living in the ocean and to reproduce you have to just release gametes into the water sort of randomly, it really helps if all the other ones around you do the same thing. Right? So if you released your gametes in May and the closest neighbor released their gametes in September, you might have a hard time reproducing. But if everybody releases their gametes on the same night, then there's going to be some successive reproduction, and that will then, kind of proliferate the next generation. So that cooperation there is also when it comes to reproduction. And then just building those big complex structures like those barrel sponges or those tube sponges does require a degree of cooperation that you wouldn't really see if it was strictly a colonial organism. All right, so the outer layer of a sponge is called the epithelial layer. This is where we find the ostia or pores. Pores and ostia are the same thing. The osculum is still technically part of the epithelium as well. So a lot of the water incurrent and excurrent function happens through the epithelium. There is a mesohyal layer, which is a lot of where you find the spicules and spongin. Not necessarily quite as many cells, but a lot of structurally important stuff that happens there in the middle layer. And then on the inside of the animal, we find the collar cells, which are the ones that actually contribute to the water circulation. The ones that are the sort of the functional foot soldiers of the sponge are the choanocytes or collar cells that we find in the innermost layer. They're going to line the internal space within the sponge, which is called a spongocyll. Spongocyll is it's not a body cavity, but it's a space within the body of the animal, which is where the water kind of comes in and then goes out from the osculum through that spongocyll layer. All right, so we've got our collar cells, and again, each one of those collar cells also has to capture and digest its own food. This is what we would consider to be intracellular digestion, because food particles are broken down within individual cells. Again, if we think of our own bodies, we haven't covered the GI tract yet or digestive system yet, but when we digest food, it's broken down outside of cells, right? We've got food in our stomach, we've got food in our small intestine, and when the food gets there, the cells surrounding that food release enzymes or other substances to break down the food, and then the cells absorb the nutrients that are released afterwards. Right? The cells don't actually bring in food and digest it within the cells. They release things to digest the food outside of themselves, and then that's absorbed by whatever kind of animal we're dealing with. And we'll talk a lot more about that as well. Sponges, we talked about only move in the juvenile stage. We talked about growth patterns. We talked about the feeding of these things with the filter feeding nature, the choanocytes, creating that water current. Water, again, going in through the ostia, coming out through the osculum. Very much protist-like. And these sponges can filter a large amount of water in a relatively short period of time. One of the reasons why, if you think about like tropical waters, Caribbean waters, Hawaii, those kinds of places, South Pacific, you always think of the water being really, really clear. And that's the reason sponges are one of the reasons why that water is so clear, is because they are filtering the water. They are taking whatever sediments, whatever bits of organic matter that might be in the water, and they are taking it out of the water and digesting it, and then the water that comes out the other side of the sponge is relatively clean and relatively clear. We see a lot of filter feeders in Caribbean and other tropical kinds of places in the ocean, which means the water is going to be really, really clear. And so that's, it's not that the water is necessarily pure on its own, it's that it's been filtered out by other kinds of organisms. This picture, by the way, is this is called a Venus flower basket. And what this is actually is not what the sponge normally looks like when it's alive. So what's been done here is these two sponges have had their spongin digested away. They were actually exposed to hydrochloric acid, which essentially denatured all the spongin and left behind all the spicules, which form kind of like the lattice work structure of the sponge itself. All this white stuff you see here, these are spicules made out of silica, or that glass-like structure. And so what's left behind then is the supportive part of the sponge when you take away the spongin, which kind of was what glues everything else together. So this is actually something that was made artificially, this whole kind of setup here, to illustrate what the inside of a sponge looks like when you take away all that spongin and when you take away the cells as well. So again, we see our picture here of the different layers of the sponge. Different kind of way of view. This one's actually showing us that intracellular digestion where the food particles have been brought in by these collar cells. These little dots are food particles. And every single collar cell has to capture and phagocytize its own food particles. If you guys remember phagocytosis, which is cell eating, it's a type of bulk transport that we talk about in Principles 1. Every cell has to phagocytize its own food and digest it within the cell itself, as opposed to what we do, where food particles are broken down outside of cells and then the nutrients are absorbed after the fact. So that's the inside of a sponge. Again, ostia, collar cells, osculum, 3 layers, mesohyl, epithelium, collar cells. Just kind of reviewing some of the structures we've seen so far. Now, one of the things that you might sort of consider as being a problem for a sponge is, number one, being grown over by other organisms, because they're not especially active necessarily when it comes to fighting off competition. You might also wonder about predation, because these things, if they're attached as adults, would just be considered like a sitting duck for any sort of fish or any other kind of organism that might come along. But it turns out there really aren't a lot of things that eat sponges, and there really aren't a lot of things that can grow over a sponge, because, number one, for predation, those spicules are a very, very good mechanical defense against predation. You're not going to find too many things that like to come along and get a mouthful of what amounts to broken glass, right? Spicules, when you bite into them, it's like taking a mouthful of broken glass and trying to consume it. So there really aren't too many things that can handle that. And for other kinds of organisms that are also kind of similar, that might want to grow in the same place as a sponge, sponges actually have some chemical defenses, so that if something tries to grow on them, they can either A, poison it, or B, just sort of like shake it off, not really like literally shake, but sort of dislodge it from the body of the sponge so that it doesn't grow over the sponge itself. So they've actually, they've been around a long time. These are the oldest animals. They've been around maybe 700 or 800 million years. Not by accident. They're pretty good at what they do. They're not a huge group, but they've been around a long time, and they will be around for a long time. They've sort of refined how they live and doing it well in a pretty good way. So they're not as easy of a target as you might expect. In terms of reproduction, there are some different variations of this. Sponges can reproduce asexually through what's called fragmentation, which is where a part of a sponge breaks off. Maybe it sort of moves with the current a little ways away, and it sort of attaches to the substrate there. That would essentially form a clone of the original individual, and that's actually pretty common. Sponges are, because they're so simple, and because they're relatively unspecialized, you can take a sponge, and even more extreme than the example I just gave you, you can take a sponge and break it apart at a cellular level, and those cells are actually able to recombine and reform the body of the sponge if you give them the opportunity. If you bring them back together, they will reform the original sponge because the cells are unspecialized. Now, if you take a more specialized animal, if you try to take your cat and separate it at a cellular level and then expect those cells to come back together, you're going to be waiting for a while, and don't do that. It'd be a very sad day if you did that. So, a more specialized animal, if you take the liver cells and separate them from the rest of the animal and then bring them back together, too late, it's not going to work, because those cells have to have the other cells to support them all the time, otherwise they're going to die. Sponges can also reproduce sexually. Some of the choanocytes can turn into sperm cells. There are cells that also will turn into egg cells. There is a lot of cooperation, again, when it comes to reproduction, to make sure that those gametes are in the water at the same time so that fertilization can take place. What we see up here in this picture is a juvenile or a larval sponge, which has cilia, which allows it to swim until it hopefully reaches a suitable substrate where it can then attach and turn into the adult form. So they are motile as the juvenile form. Juvenile sponges, by the way, are also planktonic, and this is a term we're going to see quite a bit throughout the animal section of this course. Plankton it refers to sort of anything that's small enough in the water that it's at the mercy of the water currents. Right? Things that are so small that they really can't swim against the current, and they just sort of have to go with the flow. Juvenile sponges are microscopic, and even though they can technically swim, they can by no means swim well enough to go against the water current and actually, go anywhere but where the water is taking them. So plankton really can consist of anything that's that small, anything that's at the mercy of the water currents. So in the plankton, you can find not only juvenile sponges, but juvenile stages of corals and mollusks, and even things like fishes in some cases can be planktonic if they're small enough. So very much at the mercy of where the environment takes them. So from there, we're going to move into the eumetazoans, and we'll be talking about the cnidarians first. But before we do that, I just wanted to wrap up by talking about what a eumetazoan is first, and then we'll kind of wrap up this section. Eumetazoans, or just metazoans, that works as well, are animals that have distinct tissues. Right? They have tissue-level specialization. They have cells that work together to do different kinds of jobs. So with that, with the idea of having tissues, you then have embryos that have distinct tissues as well. So in the last chapter, we talked about endoderm, mesoderm, and ectoderm, which are the layers in the embryo that give rise to the various layers in the more mature stage. So that is also something we didn't see back when we covered the sponges a few minutes ago. For example, we talked about these in the last section, but the endoderm forms the digestive tract, the ectoderm forms the epidermis and the nervous system, the mesoderm forms muscles and connective tissue and those kinds of things. So, these embryonic layers are also unique to the metazoans, and it's not surprising because the metazoans have specialized tissues that have to originate somewhere, so it makes sense, and the embryo would also have some fairly distinct layers. Metazoans, because they're specialized, also have true body symmetry. They have two equal halves. Most of the metazoans have bilateral symmetry, where there's a equal left and right side, but there is one group of eumetazoans that has radial symmetry, as we'll talk about. We mentioned them back in the previous chapter as well. But that radial symmetry is a very different kind of symmetry from what we find in most of the symmetrical animals, which is bilateral symmetry. Radial symmetry, remember, is essentially an infinite number of lines of symmetry. It's not just one like we see with bilateral, but with radial symmetry, you have animals that are essentially circular or spherical, and because of that structure, they're able to form two equal halves essentially in any direction. The radially symmetrical animals are diploblastic, right? So diploblastic, again, means that they have only two germ layers. They have endoderm and ectoderm. They do not have a mesoderm. They have something else instead called a mesoglia. Mesoglia is a gel-like structure, and if you kind of can predict where we're going with this, and we talk about things like jellyfish and corals and anemones, the kind of squishy nature of those animals is essentially mesoglia. It's the stuff that fills in the space between the ectoderm and endoderm when the animal's developing, and later on is the stuff that fills in the space around the different tissue types in the adult form as well. So with the radially symmetrical animals, there actually is only one phylum, the cnidarians, right? Cnidarians are things like the hydroids, which we'll talk about, very diverse group there, jellyfish, corals, anemones, a few other things, but those are kind of the bulk of the radially symmetrical animals within the cnidaria. The other phylum, which was historically classified as radially symmetrical, was the Ctenophora, or what's commonly referred to as the comb jellies, but these actually are not truly radially symmetrical. In recent years, they found that they do have a form of bilateral symmetry, and they do have a mesoderm in the developmental stage. So the Ctenophora historically were classified as radially symmetrical. They've been reclassified now with the rest of the metazoans as being bilaterally symmetrical, and for that reason, they're considered to be less closely related to the cnidaria than what they once were. All the other phyla, with the exception of the periphery, of course, are triploblastic and have a degree more specialization. In some cases, many more degrees of specialization. But the reason why I mention that with the classification is to show you that, even though people have been studying animals for hundreds of years and classifying them for at least, 300 years or so, this is by no means very well established, and things are constantly in a state of reclassification. As more information is discovered, animals are placed in groups that they weren't initially placed in as new information comes along. All right, so we're going to wrap up the first section here, and we'll start up with our second section in just a moment.